Perfect. All right. So, over the past couple days, or I guess it's closer to a week now, we have talked about what the cell cycle is, what the life cycle of a cell is, and we've gotten really familiar with the concept of mitosis and how a cell goes from one uh, like regular living cell whoop, into two daughter cells. So today we're going to talk about how that process is regulated. How does a cell go, or how does a cell know when it's going to divide? How does a cell know when to start division? When does a cell know when to stop division? What happens when all of that goes wrong? So, before we get too far into this, does anybody have any questions about mitosis, about cell division, about anything for my class? No? All right, if you think one, let me know. Um, we are going to start with a question. Is there some sort of signal that directs or regulates the cell cycle? This was a really big question in the 1980s, early 1980s, late 1970s. We knew what the cell cycle was. We knew how cells were to divide. The technology that we used to be able to see cells had gotten good enough so we can actually recognize what the chromosomes are doing inside of a cell. So we know that our cell cycle has a G1 phase, an S phase, a G2 phase. It goes through mitosis, prophase, metaphase, anaphase, telophase, cytokinesis. We've got that. But why? Why does this happen? How does this happen? How do cells know when to divide? So, they set out to figure out what kind of proteins and what levels of proteins exist in a cell during different phases of the cell cycle. And they found a protein called cyclin, or that they named cyclin because they're very creative. Cyclin. Cyclin is a protein that when they injected it into a cell, started division. They found this protein, they whipped a bunch of it up, they took it out into like a little needle, injected it into a cell, and that cell instantly started dividing. It is a protein that, when injected to the cell, led to division. This was a major breakthrough. We can now make a cell divide whenever we wanted to just by injecting this protein. Yes. This is for all body cells. So we're not talking like eggs, sperm. We'll get into that next semester. But like any other cell in our bodies, yes. Bacteria, same thing. So they found this one protein called cyclin, but it was actually a part of a broader family of proteins that they just called cyclins. So we have a family of proteins. So as time went on, as scientists were able to differentiate between proteins, as they were able to kind of dig deeper into the cell, they found that the cyclin protein was actually part of a family of proteins called cyclins. And they regulate the cell cycle. Bless you. In eukaryotic cells. So, all eukaryotic cells are regulated by this protein. If you remember, eukaryotic cells are like you and me. They have all of the membrane-bound organelles. So, plant cells, animal cells, and the bigger unicellular organisms. They're all regulated by this family of proteins called cyclins. They are also known as regulatory proteins, also known as regulatory proteins, or proteins that regulate something. They can control the cell cycle from inside and outside of the cell. From inside or outside of the cell. This means that there are some proteins. Yes. I have to call first. Is it like, gotta go to the nurse? Okay. Give me 1 second. Let me pause this. All right. So, like I said, we have some cells within this, or some proteins within this family of proteins that if you just put it on the outside of a cell, like if you have a culture of cells and you just kind of spread this protein out on top, it can do some regulatory actions. We have other types of proteins that when you inject them into the cell, they can have some sort of regulatory effect. So we've broken this down into internal and external regulators. Internal regulators help the inside of the cell stay on track. External regulators help the cell stay on track from the outside. So start with internal regulators. Whew. Internal regulators are proteins that a cell makes itself to help keep everything on track. They respond to the events inside of the cell. Respond to events inside the cell. So if a mitochondria isn't functioning properly, if the Golgi apparatus isn't packaging things correctly, these internal regulators will respond to that and help the cell act accordingly. They allow the cell cycle to proceed only when certain events have occurred, like checkpoints. They allow the cell cycle to proceed only when certain events have occurred. Like checkpoints. So there is a checkpoint between the G1 phase and the S phase. So the cell has to grow to a certain size before it can start replicating its DNA. So if the cell grows and grows and grows and eventually gets big enough that it can start the replication process, there is an internal signal that is made in response to getting so big. And that internal signal triggers the DNA to start dividing, or the DNA to start replicating. And then whenever the DNA is completely replicated, we can trigger the next phase where, okay, we've got enough DNA to make stuff for 2 cells. Let's start making extra Golgi and extra endoplasmic reticulum to get the next phase in the cell cycle started. And then when everything is ready, the cell has to make sure that has everything it needs to start mitosis. So there's another signal that can be made whenever we've got enough DNA, we've got enough prep done, we can start dividing. There's another signal, another checkpoint to reach. So that's internal regulators. Any questions here? Doing okay? Okay, so that's internal regulators. There are also external regulators. External regulators are proteins that respond to events outside the cell. That respond to events outside the cell. Their main goal is to speed up or slow down a cell. So if something happens and the cell needs to slow down, if we don't need a cell to grow a certain point, or like we're just done, like we've got enough, we can have these external regulators that tell the cells to, chill for a minute. Another big example is growth factors. Growth factors tell a cell to start dividing. They stimulate growth and division. This is really important in development. So going from like a single cell into an embryo, that this growth factors are really, really important. But they are also really, really important for the process of healing. So if you get a cut or scrape, right? Or if you break a bone, the body has to heal that. And they do so by releasing these growth factors. And those growth factors tell the cells on that scrape, in that cut, in that bone to start growing, to start dividing because we need more. We lost some, they were damaged. So, in order to heal our bodies, they release these growth factors, and these growth factors stimulate our cells to divide so we can get the cells that we need to heal. Important for healing. But sometimes we need cells to stop growing. Sometimes if a cell will malfunction and it will cause damage to our bodies if it continues to grow. And in that case, we get something called apoptosis. Underline that one for me. Apoptosis is programmed cell death. If a cell becomes too damaged to function, if a cell has faulty organelles, or if we just don't need that cell anymore, we can signal that cell to die. Just say, done, send signal, cell gone. What happens is it breaks up into pieces. That cell breaks up into pieces and is digested by other cells. It is digested by other cells because if a cell it has something wrong with it, right? If a cell has something bad inside of it, if it's toxic, we don't want to just release it into space. We don't want that bad stuff getting into other cells. So we have special cells that come up and like eat it, chop it up, digest it. So whatever was bad about that cell doesn't happen to other cells. But not all cells that undergo apoptosis are bad. Feel better? Not all cells that undergo apoptosis are bad. If you look between your fingers, right, you've got that little bit of webbing right there, right? Really, really hurts when you get a paper cut on it, right? But when embryos are developing, when we were just coming into fruition, our cells between our fingers or our webbing was a lot bigger. We had kind of webbed fingers. There's skin kind of connecting these like duck feet. But we want full range of our fingers. So as they were forming, as that web was forming, we said, okay, we've got enough strength in our fingers that they can stand alone. We don't need them to connect anymore. And we signaled apoptosis in those webs. Some people are born with bigger webs between their fingers because that apoptosis signal didn't come through. And that's just one example. There are a ton of examples where we need certain cells to die. That makes sense? I can't stop moving my fingers. So, but what happens if apoptosis fails? What happens if the things that regulate the cell cycle don't regulate the cell cycle? If the cell cycle goes unregulated, if apoptosis doesn't happen, that is when cancer happens. Cancer happens when body cells lose the ability to control growth. Those checkpoints we talked about earlier, they'll just blow past them. They will divide and divide and divide and divide and divide. Cancer doesn't respond to the signals that regulate the growth of cells, and as a result, divide uncontrollably. Cancer cells don't listen to the signals that regulate the cell cycle. So, when cells bypass the regular regulations, I guess, they'll just keep dividing, and that constant division is how we get cancer cells. Now, a clump or a mass of cancer cells is called a tumor, but not all tumors are cancerous, right? It's kind of one of those all squares are rectangles, but not all rectangles are squares kind of thing, where all cancer happens as a tumor, but not all tumors are cancerous. There are tumors that happen in our bodies because just other growth that's not necessarily uncontrolled. only the ones that spread are considered cancerous. So, little disclaimer, not all tumors are cancer, only the ones that spread. Sometimes our body triggers growth of cells that they think is supposed to happen, so they think it's still regulated. It doesn't necessarily spread, but that's how we get benign tumors. So that's what cancer is. And we're going to talk about some causes. What causes this uncontrolled growth? Most of the time, what triggers this is defects in the genes that regulate growth. So the proteins that are our internal and external regulators, those proteins have to come from somewhere, and they come from our genetic makeup. So if something happens, if that genetic makeup is mutated and we can't make the right regulatory proteins, then our cells won't have those regulations to adhere to. Defects in the genes that regulate growth and division. But how do these defects happen? The answer is a lot of different ways. Sometimes they're inherited. So, the breast cancer genes are a really big example of this. So, the chances that someone will have breast cancer are dramatically increased if they have this gene called BRCA. It's short for breast cancer. But I think it's the BRCA2 gene is inherited from parent to child. And if you have that gene, you are more likely to get breast cancer than someone who does not have that gene. So sometimes those defects are inherited, and other times they are caused by other things. Carcinogens, like the ones in cigarette and vape smoke, they can get into your DNA, change it, and cause uncontrolled growth. Radiation is another big example. That's why being exposed to the sun's rays, not wearing sunscreen, can cause skin cancer because that radiation can affect those genes in our skin. And sometimes, not very often, but viruses can do it as well. Because the whole point of a virus is to hijack our genetic information. And we'll get into that when we talk a little bit more about DNA. But sometimes those mutations can be caused by viruses. So we know what cancer is. We know how cancer happens. How do we treat it? How do we use what we know about uncontrolled cell growth to counteract it? Well, one way is to just remove all of the defective cells. If a cancerous tumor is contained and hasn't started spreading yet, if we can get all of those uncontrolled dividing cells out, then there's not going to be any left in there to divide. So if we can remove that tumor and get all of the cancer cells, that's a good way to do it. Another option is radiation. My mother had thyroid cancer when I was little, and radiation was the main way that they treated it. So they got most of it out uh, with removal, but radiation will destroy. Okay, sorry, got a little ahead of myself. Radiation, we talked about, mutates cells, right? It's one of the causes for cancer. How do we use one of the causes for cancer to defeat cancer, right? The answer is how it affects our DNA. So radiation destroys DNA, particularly in quickly dividing cells. So we can target the cells around where that cancer bit happened. And if a cell is dividing very quickly, it's more likely to make mistakes in its DNA. If we can increase that mistake rate, then we hope that our bodies can see that this cell's DNA is damaged beyond control, and or that it doesn't have the ability to like make things that are important for the cell's survival, so it dies. And then the third option is chemotherapy. Chemotherapy also targets rapidly dividing cells. Chemotherapy targets rapid dividing cells. We know that cancer cells divide quickly. So if we can find a way to attack the cells that are dividing quickly, we can hopefully attack all of the cancerous cells. This is why people who undergo chemotherapy lose their hair, because hair cells are quickly dividing cells. Where your hair is growing out of your head, those cells are dividing very quickly. So that the chemotherapy attacks that because it can't differentiate between cancer cells and regular cells. It just knows fast dividing, kill it. Same thing with stomach issues. So people who undergo chemotherapy have a lot of stomach issues. That is because these cells that line our stomachs also quickly divide. They have to quickly divide, or else the acid in our stomachs will cause us pain. So that's another reason. But getting the cancer cells out is better than the side effects that happen. So I know this is serious stuff, but understanding the underlying processes, understanding how this happens and why it happens helps us protect people. So there are still people who have dedicated their lives to helping create ways to destroy cancer cells. And the more we understand about cancer cells, the more we understand about how cells divide, the better we can get at treating it. And that's why we learn about this stuff. Because understanding the ways that our cells work help us understand when stuff goes wrong. Anybody have any questions, comments, concerns? There's an exit ticket on Canvas if you could fill that out for me, please. It has to do with interpreting a graph. So if you have any questions with that, please let me know. And if you haven't turned in your mitosis assignment from last Friday or the virtual lab from yesterday, try to get those in ASAP.